Assalamualaikum and hi everyone. My name is NAJIHAH binti Muhammad SYAZILI. Today, I want to carry out an experiment regarding to the rate of photosynthesis. The objective of this experiment is to determine the effect of light distance on the rate of photosynthesis. Introduction to photosynthesis. Photosynthesis is the process which plants and other photosynthetic organisms use the energy captured from light in the form of NADPH and ATP to convert atmospheric carbon dioxide into carbohydrates. Photosynthesis occurs in chloroplast, where it found mainly in mesophyll cells. Chloroplast contains photosynthetic pigment substance that absorb visible light energy. Two stages involved in photosynthesis, which are light-dependent reaction and light-independent reaction, or called Calvin cycle. Light-dependent reaction take place in the thylakoid membrane and require a continuous supply of light energy. Chlorophyll absorbs the light energy, which is converted into chemical energy through the formation of two compounds, ATP and NADPH, as electron carrier. In this process, water molecules are split and release oxygen as product. Calvin cycle, also called the light-independent reaction, takes place in the stroma and does not directly require light. Instead, the Calvin cycle uses ATP and NADPH from the light-dependent reaction to fix carbon dioxide and produce 3-carbon sugar glyceraldehyde 3-phosphate or G3P molecules that will form glucose. So, what is the hypothesis of this experiment? The hypothesis is when the light intensity increases, the rate of photosynthesis also increases. Take a species of plant and by using a hole punch, get a 10 leaf disc from the plant that I choose. Remove the plunger from the string and remove the cover from the tip. Put the leaf disc into the barrel of the string and tap them down to the tip. Take a beaker and mix with 0.5 grams sodium bicarbonate with 150 ml water. Draw about 6 to 8 ml of bicarbonate solution into the string. Hold the string with the tip up and expel the air by gently pushing on the plunger. Plug the tip of the string tightly with finger and gently pull on the plunger to create a slight vacuum. Repeat the previous step several times until all of the discs have sunk to the bottom of the solution. When all the leaf discs have settled to the bottom of the solution, carefully remove the plunger and pour the disc and solution into the cup. Set up the light so that it is suspended about 3 cm above the beaker. After several minutes, the disc should begin floating to the top of the solution. Record the number of floating discs every minute until all discs are floating. Repeat the step before using the same leaf, but this time set up the light so that it is suspended about 10 cm above the beaker. Put the last cup in the dark for 15 minutes. As a conclusion, photosynthesis will occur in the presence of light energy, carbon dioxide, and water. This experiment also proved that when the light intensity increased, the rate of the photosynthesis also increased. It has shown when the light source is put near to the beaker, all the leaf disks are floating faster compared to when the light source was placed far from the beaker.